Hello, everybody. I'm Seda Kurt from the International Biogeography Society, and welcome to the TIBS Funk Lecture Series on Biogeography. We're pleased to welcome Dr. Barnabas Daru today, and his talk is about the biogeography of native botanical diversity in the age of humans. Now, as we wait for people to show up, let me go over some information on upcoming TIBS events. First of all, I'd like to emphasize that we now go by the new acronym TIBS, and we also have a new logo. And the next funk lecture, Biogeographic Perspectives on Amazon Forest Degradation by Dolores Armentes Pascual, will take place on May 29. Registration will open next week. The TIBS Biennial Conference will be held in Aarhus, Denmark. You can check the TIBS website for recent calls and updates that will be announced soon. And after Dr. Daru's presentation, there will be a 20-minute Q&A session. Please leave general comments on chat. And if you have any questions, type them in Zoom's Q&A section or ask them live by raising your hand and unmuting yourself once you have received permission from the system. Lastly, let me welcome you to Dr. Marta Yajina, the new TIBS VP for Public Affairs and Communication. She'll be in charge of organizing the funk lectures. So please, Marta.
Thank you very much, Seda. Welcome, everyone. And as Seda mentioned, my name is Marta Yajina, and I have recently been appointed as the Vice President for Public Affairs and Communications here at TIBS, taking over from Sandra Noak. And in my first task as the member of the Board of Directors, it is my immense pleasure to introduce today's funk lecture series speaker, Dr. Barnabas Daru. Barnabas Daru is an assistant professor of biology at Stanford University, and broadly speaking, Dr. Daru's research focuses on the ecology and biogeography of plants across ecological scales. His research group often uses herbarium specimens and phylogenies to understand how species are distributed across the world, what are the underlying drivers of these distributions, and how these distributions might change in the Anthropocene. Barnabas was first drawn to biogeography as a child when he spent hours exploring the volcanic mountains near his hometown in Kerang, Nigeria. He went on to complete a bachelor's degree in zoology with honors from the University of Jos and a master's and Ph.D. in ecology and evolutionary biology from the University of Johannesburg in South Africa. In his doctoral work, he identified new biodiversity hotspots for woody plants in southern Africa. And following his PhD, Dr. Daru pursued a postdoc at Harvard University, where he explored the unrealized potential of herbarium collections for addressing questions in ecology and evolutionary biology. In his most recent work, Dr. Daru assembled an unprecedented global database of native plant distributions, which includes range maps for over 200,000 plant species. And using this database, he quantified the impact of climate change on plant biogeographic boundaries and explored biotic interchange of plants in historical and contemporary times. So despite his relatively young academic age, Dr. Daru has already become a prominent figure in biogeography. He's the author of more than 45 scientific publications in top-tier journals, including Nature, Nature Communications, and PNAS. He's also a passionate educator and mentor, and he is committed to promoting biogeography among people from historically marginalized populations. In his free time, he enjoys swimming and hiking with his family, and exploring nature that he loves so much. So we are very, very excited to hear about Dr. Daru's fascinating work. And without further delay, I turn it over to Barnabas.
Okay, thank you very much, Martha, for that kind introduction. And thanks, everyone. And thanks for inviting me to talk about my research, which I entitle The Biogeography of Native Plant Diversity in the Age of Humans. So I'll start with a brief introduction of this musician, John Legend. He is a very popular musician and often considered a solo artist. And he is well known such that his net worth is over $100 million. And he lives in Beverly Hills, California, in the USA. However, whenever he is on stage, I often consider him not to be a solo musician per se, because if you zoom in closely while he performs on stage, just like many other solo musicians out there, you notice that there are other people in the background as well as even in the backstage. And so these other people who are not often the center stage of the performance are playing key roles in the music to really contribute to making the music sound really beautiful. And so these people, for example, there's a person, behind the scene here playing the guitar, another person playing the trumpet, and yet another person playing another instrument, while someone who is not even shown here is playing the drums. And so these people are really important. And so without them, this music will not really see the light of day. And so I consider these other people who are hidden behind the scenes as the unsung heroes of music. And so while we know about, popular musicians like John Legend, we do not really know much about these other musicians in the background that are really contributing to making this sound really beautifully. We don't even know their names, let alone we don't know where they live. And so in my view, I consider plants as the unsung heroes of the ecosystem. They are so important because they provide food and medicines. They also regulate ecosystem processes. And in fact, the whole biosphere depends on plants being primary producers. Yet, we do not know the geographic locations of many plant species across the globe. And so our research is a step in this direction and seeks to understand the processes that generate, distribute, and maintain plant diversity. And so for today, I'll talk about 3 questions. So first, I ask the question, where are species distributed today? And how do we determine what unique communities to conserve? And what are the mechanisms that drive biodiversity change in the Anthropocene, especially in this area of profound human impact on the environment? And that's how I've outlined my talk today, starting with understanding patterns of species distributions and ending with how to understand biodiversity change in the Anthropocene. For the first question, where are species distributed today? So I'll use a case study that we looked at predicting cryptic native plant diversity at a global scale. Now, plants, like I said, are very diverse and occur in different kinds of environment, ranging from forests to savannas, and they even occur in extreme environments such as the Arctic tundra, and there they maintain ecosystem processes. And over the years, botanists all around the globe have also made tremendous efforts in trying to locate and document the distributions of plants across the globe. And this is an example of our own effort along these lines as well, where we explored and aim to document the distributions of plants in different places around the globe, including the Miombo woodlands, the Cape Floristic region of South Africa, Kruger National Park, or Alaska in the Arctic regions. And so consequently, we brought these plants back to the herbarium and we prepared vouchers of these specimens. And subsequently, we also image them and digitize them, making them available online for for more wide accessibility. But yet there is another kind of data set that is really gaining momentum, and this is derived from citizen science projects. And these are often observation-only records derived from mobile applications and citizen science projects. And so put another way, I can consider that biodiversity data can be grouped into two types. Voucher specimens, that's those that are often considered primary data, which have links to a tangible material in a museum or herbarium. And on the other hand, there is observation-only records. These are often sightings and do not necessarily have a link to a physical material in a museum or herbarium. But together, these data sets can contribute to informing our understanding of biodiversity across the globe. However, though, these data sets, because they were non-randomly collected, they also have biases in them. And so along with my student, Jordan Rodriguez, we explored coverage gaps and biases in plant occurrence records. And so we compared how well voucher specimens document our expected knowledge of biodiversity compared to observation records. And so in this map, I show here the expected coverage of species richness as documented by voucher specimens, such that areas in darker colors correspond to high coverage and lighter colors correspond to low coverage. And I contrasted that with observation-only records. And we found that while there are massive data gaps, especially in places like that are known to concentrate high biodiversity, such as the neotropics or the Afrotropics of Southeast Asia, voucher specimens do capture expected knowledge of biodiversity way more than observation-only records, suggesting that the quality of voucher records are way more than observation, despite both of them do have biases in them. And so we propose that we can use species distribution models to overcome these biases. And I'll walk you through briefly an experimental framework that we use to achieve this. So first, we explored and sourced all the possible data sets on the known occurrences of plants across the globe. We subjected these data sets to several cleaning and filtering steps, and eventually we generated polygon maps from these data sets, and we used that to define a calibration area as well as a dispersal rate to constrain our predictions based on the species distribution model. And this calibration was based on a phylogenetic framework to define the dispersal rates for the species distribution modeling. And then we also generated background points as a function of the training area as well as the sampling biases in plant occurrence records. And then we combined these different data sets, the point occurrences, the training areas, as well as the background points in a species distribution modeling framework to generate range maps for over 200,000 species at the finest scale resolution possible, 9-kilometer resolution. So we use these data sets and make the following hypothesis. So we hypothesize that if our current knowledge of plant sampling indicated here in yellow is biased and thereby limiting our knowledge of predicting true native plant diversity, then we hypothesize that we can use machine learning tools as a function of unbiased data, for example, tetrapod diversity, sampling intensity, as well as other variables that are known to predict plant distributions across the globe to predict true native plant diversity. And so when we use this framework, we then address the following questions. So first, we ask, what are the key patterns of modeled vascular plant diversity? And we also ask, how can machine learning validate these patterns and uncover refuges of hidden plant diversity? And how effective are protected areas in capturing plant diversity centers in need of conservation efforts? And when we did that, we found some really exciting results. So first, we mapped global patterns of native vascular plant diversity. An indicator here in this map is species richness, which corresponds to the count of species within 9-kilometer grid cells. Areas in darker green corresponds to high richness, and brown areas correspond to lower richness. And so you'll notice that we found unique patterns of plant diversity, especially in undocumented biodiversity-rich regions. And when we even contrasted our results along a latitudinal gradient and contrasting that with latitudinal diversity gradient for tetrapods, which includes birds, mammals, amphibians, and reptiles, which are generally well known than plants, we found strong latitudinal gradient that support increasing diversity toward the equator. However, we also explored the congruence of expected plant species hotspots compared to those of tetrapods. And so here we overlaid a hotspots map of plants indicated here in deeper red on top of those of tetrapods indicated here in darker shades of gray. And so areas of overlap of plants and tetrapods are indicated in lighter red. And so we found that plant hotspots weakly overlap tetrapods in some areas than even within tetrapods groups themselves, suggesting that plants do capture other facets of diversity that cannot be predicted based on tetrapods alone, and also suggesting differences in the evolutionary history of plants compared to those of tetrapods. We then, to validate our results, we use a machine learning extrapolation based on random forest, and we assume a globally high sampling in the future. And so when we train our models based on these machine learning algorithms, we found that 43% of diversity hotspots will be robust to sampling effects, especially as indicated here in these areas in purple, suggesting that our random forest extrapolation is able to validate our model estimates by this much. But however, though, we also found that we uncovered previously undetected hotspots indicated here in teal that represent an increase of 57%. And so these undetected hotspots correspond to places that are not currently discovered based on current sampling effort. But if sampling intensity is increased in the future, then these are places that we stand to discover hidden plant diversity. And so these could represent areas to target for future collecting, as well as to prioritize conservation efforts. Then next, we explored the coverage of these diversity hotspots within current network of protected areas by overlaying this hotspots map onto existing protected areas. And in this map, areas in red corresponds to hotspots that are falling outside of protected areas, whereas darker colors are hotspots that are inside protected areas based on the model estimates. And we found that only 35% of hotspot cells intersect with protected areas. The pattern is even much more pronounced when we looked at the extrapolated hotspots based on random forest extrapolations, finding that more than 60% of the extrapolated hotspot cells are outside current network of protected areas and vulnerable, suggesting that conservationists should expand protection to capture these additional facets of diversity that are lying outside of protected areas. But we also tested whether it's worth extending protection to these unprotected hotspots. To do that, we explored coverage of other species level attributes, including plant height, seed size, evolutionary distinctiveness, and extinction risk within hotspots that are inside, as well as those that are outside protected areas, indicated here in areas below the zero line. And so we found that while protected areas are doing a great job in protecting species that are generally larger in size, as well as those that are at elevated risk of extinction, we found that unprotected hotspots do harbor species that are evolutionarily distinct, meaning species that do have fewer or no close living relatives, suggesting, and examples of these species are species such as eremosine or Aricaceae that are found uniquely on the phylogenetic tree of life of plants and currently lying outside of protected areas, suggesting that it is indeed worth extending protection to capture these facets of diversity that are lying outside of protected areas. So going back to the initial question in terms of where are species distributed today, so I showed how model estimations uncover unique patterns of native plant diversity with similarities and differences to those of tetrapods. I also showed how machine learning can identify undetected native plant diversity, especially in biodiversity-rich but undersampled regions. And lastly, we showed how most plant hotspots are currently unprotected but do have high evolutionary distinctiveness and in need of protection. So going back to the initial framework in terms of how we seek to conserve unique communities, so pose the following question. So how do we determine what unique communities to conserve? So here I'll talk about a research project that I did back in my PhD on phylogenetic regionalization of Southern African vegetation zones. So Southern Africa is a very diverse and heterogeneous landscape with different kinds of vegetation ranging from deserts to evergreen forests, and is also floristically diverse with over 24,000 species of vascular plants recorded in the area, such that if you sort of explore the landscape of Southern Africa, starting from the southernmost tip of the continent of Africa, you find this Mediterranean type biome, also known as the Cape floristic region, with high species diversity and endemism. And then further inland, you find the savanna, which is one of the defining features of the African landscape, often characterized by woody vegetation as well as grassland. And so along the eastern seaboard, you have the Indian Ocean coastal belt. So the question is, how were sort of the boundaries of these different biomes defined? And so to do that, we use woody species. And so we define woody plant species as mostly trees, such that other life forms like some tree aloes and proteas were included in our definition. We also a woody plant should be characterized by an above ground permanent trunk or stem, and it should be characterized by growth habits such as trees, shrubs, palms, some tree ferns, and geoxylic suffrutices were also included in our definition. And then we sought out to collect plants and woody plants along different areas in this landscape in Southern Africa. We collected herbarium specimens as well as DNA plant samples preserved in silica gel for DNA extraction. So using these data sets, we generated a phylogenetic tree of all the woody species in the region, as well as also generated richness map, geographic distributions of all the species in the region. And here in this map, areas in red correspond to high richness and blue corresponds to low richness. So combining these data sets, the phylogenetic tree of the woody species along with their geographic distribution and a metric called Simpson's phylogenetic beta diversity that quantifies the amount of phylogenetic branch lengths shared between two communities, then we regionalize based on the shared evolutionary history of the flora. And when we did that, we found 15 phylogenetically delimited bioregions, which we call phyloregions because they are phylogenetically delimited. Here, the colors in the map, the NMDS ordination plot in B, and the hierarchical dendrogram of dissimilarity in C, the colors are similar and they indicate the level of differentiation of the flora in each phyloregion, such that phyloregions that have similar colors means they have similar plant clades, and those that have different colors means they differ in the plant clades that they enclose. And so we found that our phylogenetically delimited phyloregions do correspond to the existing vegetation zonation, i.e. the biomes of Southern Africa that were defined based on climatic composition as well as expert opinion. For example, our Fynbos phyloregion do match to the existing Fynbos biome of Southern Africa. Our savanna phyloregion do match to the existing savanna biome of Southern Africa. However, we discovered other new sort of phyloregions that were not included in previous delimitation, one on the east and one on the west, which we named them the Zambesian Transition Zone and the Gariep Karoo, respectively. And so looking closely at these newly discovered phyloregions, the newly discovered Gariep Karoo in the west, we found that these phyloregions share close phylogenetic relationships to the succulent Karoo, and it has several woody species, including Allodichotoma and Pachypodium namaquanum, that are of high evolutionary distinctiveness and endemism and contributes to this area's rich evolutionary history, thereby deserving that this area should be recognized as an important plant biogeographic region. And so looking also at the second newly discovered Zambesian Transition Zone, we found that this region shares close phylogenetic relationship to the Indian Ocean coastal belt in the eastern seaboard, and it also has more than 800 woody species in the region that contributes to the area's rich evolutionary history. Lastly, we also explored the amount of evolutionary distinctiveness encapsulated in each of these phyloregions. And so we found that the Cape floristic region emerged as the most evolutionarily distinct phyloregion in Southern Africa. And so looking closely at this phyloregion, it turns out also that the Fynbos of the Cape floristic region is one of the world's biodiversity hotspots, characterized by high degree of endemism and richness. And so suggesting that there may be other processes that are structuring the diversity and endemism of this flora in the Cape region to emerge as the most evolutionarily distinct phyloregion. So extending this concept to a global scale, especially given that biodiversity patterns are also scale dependent, we expanded our analysis of phyloregions at a global scale, this time around going into the marine environment for seagrasses. Now, briefly, seagrasses are not grasses. However, they are monocots, and they belong to the order Alismotels. There are 7 families and 72 species worldwide distributed along the coastlines of the world. And so these are examples of common seagrasses. On the left here is the eelgrass, or Zostera marina. In the middle is Cymodosia rotundata. And back in 2016, seagrass made the cover of Nature, where the genome of Zostera marina was reported. However, though, apart from this exciting statistics, seagrasses also play key crucial roles in the marine environment. They are primary food for marine herbivores, such as this endangered green sea turtle that grazes exclusively on seagrasses. Seagrass meadows are also important nursery grounds for fish and other marine invertebrates. And so most of marine life depends to some extent, directly or indirectly, on seagrasses. And so to understand how seagrasses assemble into biogeographic regions, so we generated phylogenetic tree based on DNA sequences for seagrasses, and combining that with the known geographic distributions of seagrasses across the globe, where here on the right, areas in red corresponds to high richness, and blue cells corresponds to low richness. And we again combine this with the metric of Simpson's phylogenetic beta diversity and regionalized based on the shared evolutionary history of seagrasses. And when we did that, we found 11 phylogenetically delimited ecoregions, which we call them phyloregions as well. But importantly, we found that seagrass biogeographic regions or phyloregions can be grouped into a temperate as well as a tropical region, such that we found, based on our phylogenetic analysis, that geographically disjunct phyloregions do tend to harbor closely related species. And this was really exciting because it shows the power of phylogenetic approach in capturing some of these patterns that may indicate perhaps a history of long distance dispersal or historical extinction that may be structuring the diversity of seagrasses within these ecoregions. For example, explaining why species such as Zostera marina tend to distribute widely across the seascape. And when we looked at the evolutionary distinctiveness of seagrass phyloregions, we found a latitudinal gradient of higher evolutionary distinctiveness in the temperate regions as opposed to the tropical regions, which contrast to findings that were patterns that were found for terrestrial tetrapods. And so possible factors can explain this. So it's possible that climatic barriers might be limiting the exchange of taxa between phyloregions, or it could also be that tropical species with temperate origins may have been lost through historical extinction, and thereby leading to the pattern of this high evolutionary distinctiveness that we are seeing in temperate versus tropical regions. So going back to the initial question in terms of how do we determine what unique communities to conserve? So here I showed that phyloregions do match very well to existing vegetation zonations, such as the fynbos of the Cape Floristic region. And I also showed that phyloregions do differ in evolutionary distinctiveness with underlying processes. And I showed for seagrasses at the global scale that geographically disjunct temperate seagrass phyloregions can harbor closely related species. So going back to the initial framework in terms of how we seek to understand biodiversity change in the Anthropocene, I pose the following question. So what are the mechanisms that drive biodiversity change in the Anthropocene? And here I'll use a case study of the homogenization of plant communities in the Anthropocene. And this is work that was done in collaboration with an amazing team of researchers from across different institutions and different parts of the world, and which I'm really grateful for. And so I'll take you briefly on Earth's history the last 540 million years ago, such that indicated in the asterisk at the bottom are the mass extinction events, and zoomed out at the top is the last 23 million years, with the different epochs indicated: the Miocene, the Pliocene, the Pleistocene, the Holocene, and another epoch that is under question, with all the megafauna that radiated during those times indicated in the icons. And so this other epoch that is under question is beginning to be recognized as the Anthropocene. And this epoch is so defined because during this time period, we are witnessing high human population growth. The climate is changing rapidly, and species are also going extinct at an unprecedented rate. And at the same time, species are being exported and introduced to environments that are outside of their native ranges. And so for the purpose of my presentation today, I'll focus on these last two processes: species extirpations and species naturalizations, and how these two processes can combine together to drive homogenization. And so biotic homogenization is the process by which naturalization and extirpations can drive homogenization by reducing this composition of species and phylogenetic branch lengths that are shared between regions, and this process can diminish the regional distinctiveness of biotas and are likely to have negative impacts on key ecosystem functions. And so to understand how biotic homogenization may be impacting plant communities, so first we use our newly curated data set of plant species distributions at the species level for over 200,000 species around the globe, and combining this with a phylogenetic tree of all the vascular plants. And using this, then we explore changes in alpha diversity. And to do that, we took the differences between alpha diversity in the Anthropocene versus alpha diversity in the Holocene. And just to refresh your mind again, our definition of the Anthropocene versus the Holocene are these two time epochs, meaning that species composition before any form of naturalizations and extirpations corresponding to the Holocene, and species extirpation after species composition after naturalization and accounting for recent extirpations corresponding to species to the Anthropocene. And so we also did the same for beta diversity that evaluates the differences in species composition across sites for the Anthropocene as well as the Holocene. And then we explored the exchange of non-native phylogenetic diversity. So when we did that, we started by first mapping the distributions of native vascular plant alpha diversity in the Holocene. And here, areas in red or lighter colors correspond to high richness, and darker areas correspond to low richness. And so we contrasted that with alpha diversity in the Anthropocene, and that's after accounting for recent introductions as well as recent species extirpations. And when we took the difference, here areas in blue corresponds to decreases in alpha diversity, and lighter areas correspond to increases in alpha diversity. So we found widespread patterns of decreases in alpha diversity across the globe. And so when we then explored how these patterns may differ based on species composition, we then looked at beta diversity. However, because beta diversity decays with geographic distance, so we quantified beta diversity within what's referred to as the botanical biomes, also known as the taxonomic diversity working group level 3 biomes that correspond loosely to country borders. And so we looked at differences in species compositions across grid cells within these botanical biomes. And we used that to map beta diversity in the Holocene, and that's before any form of recent species introductions and recent extirpations. And we contrasted that with beta diversity in the Anthropocene after accounting for species introductions and extirpations. And when we took the difference, here areas in blue or negative values corresponds to losses in beta diversity that may suggest homogenization, whereas areas in lighter shade or red corresponds to increases in beta diversity or a process of differentiation. And so we found widespread homogenization of plant communities in the Anthropocene. And some of the areas that are experiencing the highest losses in beta diversity are places like Northern Africa or the circumboreal regions, as well as places in eastern part of Australia. And so we then explored how these patterns may be influenced under different scenarios of biotic change, starting for a scenario of considering only naturalizations only, or to a worst case scenario when we assume currently threatened species are to go extinct. And so we found that changes in plant communities are driven by non-native naturalizations than native extinctions. And even when we assume a worst case scenario such that currently threatened species are assumed to go extinct, we found that non-native naturalizations are by far the major contributors to biotic reorganization. And so looking at these process of naturalization much closely, we then look at the directionality of the exchange of these non-native species across regions. For example, species that are native to North America but being introduced to other continents of the world, or species that are native to Afrotropics and then being introduced to other regions of the world. And we did the same for all the other continents across the globe to explore the directionality and the magnitude of the exchange, such that the thickness of the lines are proportional to the amount of species that are being introduced, as well as we looked at the amount of phylogenetic diversity that is also being exchanged across regions. And even when we corrected phylogenetic diversity with species richness, across regions, we found that Asia and North America are the major sources of non-native species. However, the species that they export tend to be phylogenetically close to the recipient floras. Australia, the Pacific, and Europe, on the other hand, contribute fewer species to the pool of non-native species, but they represent a disproportionate amount of phylogenetic diversity. Going back to the initial question in terms of what are the mechanisms that drive biodiversity change in the Anthropocene, so I showed how, regardless of the extinction scenario, the strongest contributors to biotic homogenization is driven by non-native naturalizations. Asia and North America are major sources of non-native species, but the species that they export are phylogenetically close to recipient floras. So our results kind of demonstrate the profound influence of humans on how humans are exerting on regional biotas beyond just changes in species richness alone. So putting all this together, so far, I've talked to you about how our research seeks to understand where species are distributed today and how do we identify unique communities to conserve, and how we seek to understand the major drivers of biotic change in the Anthropocene. So for future and some of the ongoing research projects in the lab, we have three major projects, or most of the projects can be grouped under these three headings. So first, we are exploring ecological interactions of plant microbial symbionts, especially targeting fungal endophytes for most of the plant species that we are currently exploring. And we are also looking at developing computational infrastructure for addressing some of the questions that we are so passionate about in the area of macroecology and biogeography. And we also have projects that we're looking at macroecology and biogeography in the Anthropocene. And so I'll touch on briefly on each of these projects. So for the first project, which is also funded by the U.S. National Science Foundation, it's a multi-institutional grant to study endophytes, or microbial communities that are interacting with plants in the Arctic. So this project is led by a current student, Paul Markley. And so we are exploring the interactions of plants and microbial symbionts and using herbarium records to track the history of biotic interactions over time. And so we have done some really extensive field collecting so far in Alaska and other Arctic biomes, such as Nunavut and Iqaluit in Canada. And we're using these data sets and combining that with herbarium collections spanning the last 100 to 200 years to track the history of biotic interaction through time and how these interactions may be responding to climate change. And another project that we are also looking at is to characterize climate change and shifts in microbial symbionts, this time for marine plants. And so while we know a little bit about interactions of plants and microbial communities in terrestrial environments, we know so little about seagrasses or plants that live in marine environments. And so for this, along with student Michael Young, we're exploring microbial interactions for seagrass communities and mangroves especially in North America, to explore how these marine plants may also be responding to climate change as a result of their interaction with microbial communities. Then for the second aspect of our research, we also invest a lot in developing computational infrastructure for macroecology. And here, postdoc Colin has been embarking on this amazing project where he is looking at mapping the biogeographic regions for the world's butterflies. And so these are preliminary findings, based on, species level distributions for butterflies. And what he wants to do is to explore how the biogeography of butterflies also mirror those of plants, especially because butterflies do interact with vascular plants to a higher extent, especially the larval forms in herbivory, whereas the adult forms, play key roles in pollination. And thirdly, we are also exploring how climate change may be impacting the boundaries of plant biogeographic regions. And along with undergraduate student Samuel Bezenkri, we are exploring how climate change may be impacting the communities of the biogeographic boundaries of plants that have taken millions of years to form, but there are concerns that recent climate change may be impacting these biogeographic boundaries. And so ultimately, putting all this together, our goal is to contribute to elucidating the mechanisms by which biodiversity is generated, distributed, and maintained in the Anthropocene. And these are the current members of my lab indicated in the picture here, as well as recent collaborators and funders, as well as lab alumni, for which without these individuals, these research projects would be almost impossible. And I'm really grateful for them. And thank you so much for listening as well.
Thank you very much, Dr. Daru. It was an amazing presentation. Please put any questions that you might have in the Q&A box, and I will read them off to Dr. Daru as we go. But while we're waiting for that, I have a couple of questions on my own, maybe to get things started.
Absolutely.
I really enjoyed listening about your study of comparing distributions of plants in the Holocene with distributions of plants in the Anthropocene.
Right.
And having worked on some paleo data as focused on small mammals, so it's a little different, I know how difficult it is to actually infer what the distributions were in the past. So I'm curious, where did the data come from to map past plant distributions? Is there a database that's available? Is it all based on the fossil, on the pollen record? How did you know where plants were in the past?
Yeah, absolutely. Thank you for that great question. So while the ideal way would be to validate, fossil records do provide us with, the direct evidence of the past, we however relied on proxies in this context. And so our definition of the Holocene versus the Anthropocene were based on, simply considering, the plant composition without any form of recent naturalizations, as well as recent extirpations. And then the Anthropocene is when we then consider recent naturalizations and recent extirpations. So species composition with naturalization and extirpations, or species composition without these form of naturalizations or extirpations. And so those timelines in our context here was not really, cannot really be pointed to a specific date per se, but we consider that, the Columbian exchange, which corresponds to 1491, would be like the date on which the Western Hemisphere united with the Eastern Hemisphere, and that's when exchange of biodiversity and other things happen. And that's when food, crops, diseases, and other things were exchanged across continents. And so that may correspond to the time that species are now able to like move across continents with the aid of humans, as opposed to their natural abilities to disperse. And so that was our definition of Holocene versus the Anthropocene.